this video we will discuss about the comparison of this PCNA expression in the healthy and cancer patients. So we will discuss the PCNA expressions in the exosomes that are released by the cells, either cancer cells or the healthy control cells. In the previous video we have understood that the PCNA has been expressed. PCNA is one of the proliferating marker that expressed in different cancer cells and is a probable candidate for the universal biomarkers to detect onset of cancers at the early stage. And in this video what they did is so we will discuss some of the results that are presented in the paper. So in the figure that is on the left hand side, what they did is they first check the healthy controls, the presence of PCNA in the healthy controls, and they did this patient studies in the 26 different patients, and I is the control, that is the mixture of I is the control, that is the mixture of 3 different healthy controls. And here what they did is they checked the sera of the patients, and they are able to see the presence of PCNA in all the healthy patients or in all the healthy controls or healthy individuals who have participated in the studies. And they also checked the presence of PCNA in the patients who are affected with the cancers, and I is the control, which is the combinations of the sera of the 3 different individuals, healthy individuals. And we can see that compared to the I, there is a high amount of signal, there is a increased signals for the PCNA in the patients who are affected with the cancers in almost all the cases. And when they quantify these observations, that is PCNA in the healthy patients and the cancer patients, they are able to see a statistically significant difference, that cancer patients has high expressions of the PCNA compared to the healthy controls. And when they quantified it to find out the threshold value, they are able to find out that it's around 97.81 nanogram per ml. So if this PCNA expressions are present in more than 97.81 nanogram per ml, then there is a more chances that the patient is undergoing some kind of cancer progressions. And in order to validate this value, they did the sensitivity versus specificity analysis to find out the true positive and true negatives values, and they are able to find out that it's around 77% specificity and at around 94% approximately sensitivity, the values of this 97.81 nanogram per ml. So if this value is accurate to a specificity of this approximate 77% and sensitivity of this 94%. So actual values are presented in the paper, you can refer to that paper. There is some critical review of this paper that I have found out is that the patients that they have analyzed, the PCNA, cancer patients that they have selected for their studies, they did not mention at what stage these cancer patients are at, whether these cancer patients are at late stage or at the early stage. Because if they are at the late stage, then there might be a possibility that the patients who are at the early stage does not have significant difference of this PCNA between the healthy cancers and between the healthy individuals and the patients who are undergoing onset of cancer progressions. So these things are not mentioned in their paper. And also the studies that they did of around 300 different patients, here also they did not mention the stage at which the patients are at, whether they are at late stage or at the early stage, because that significantly affect their values or the threshold values that they find out. This could be the universal biomarkers, but there are several questions that need to be answered before someone can unambiguously say this statement. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.